Hi, everybody, and welcome back. So we're going to cover a really important part of our genes to proteins consideration today. So please watch carefully and make careful note so that you understand exactly what I'm explaining to you. There are, however, any number of other videos available on YouTube and numerous sources of information which will help you if you get stuck anywhere along the line. So we finished off in class by discussing how DNA is structured and then how it is replicated. One of the most important functions of DNA is to serve as a record of all of the proteins in the cell. DNA contains the recipe for all the proteins. And those recipes have to be handed on completely correctly every time a cell divides. So whether the cell is dividing simply as part of binary fission, as in the bacteria, or if cells are dividing in order to perform sexual reproduction, DNA must be replicated correctly, but not so correctly that evolution cannot take place. Every now and again, errors will creep in. Most of those errors are either deleterious or have no effect. But just occasionally, those errors, those changes in the DNA sequence are beneficial. And that is the stuff on which evolution is built. So let's just run over the processes of DNA replication. And this is, I'll use this diagram, which you haven't seen before, but it is exactly the same as the simpler diagram that you saw earlier. So first of all, the DNA consists of a double strand of matching base sequence. So if I have CAT there, I'm going to have ATG that way, okay? On one strand is matched by a base pairing on the other. So each strand is a template for the other strand. If I separate them, I can recreate the missing strand quite easily. That is exactly what the cell does. First of all, the DNA strand is opened. The base pairs are severed from one another, and that is done by an enzyme called helicase. And that opens up a fork, which we call the replication fork. And the replicator realized that the replication fork in this diagram is running in this direction that way. So behind the fork, we've got two strands of naked DNA. And that is protected by a set of proteins, which serve to stabilize the DNA strands and to protect them from digestion by the cell's protective mechanisms. Then an enzyme called DNA polymerase runs along the strand to replicate the bases. So if I've got a G there, it will put in a C. Sorry, let's do this one. A G there, it'll put in a C there. If I've got a T there, it'll put in an A there, etc. Now that replication cannot just start spontaneously. The replication has to start with a short length of RNA, which is laid down right at the very start, and that's called the RNA primer. And that RNA primer, once it's laid down, the DNA strand can begin to be laid down, and the RNA primer will eventually be digested out and replaced with DNA to complete the strand. So that is very easy to understand. This is called the leading strand, and it's the leading strand because it's going in the same direction as the fork is opening. At the same time as the helicase is busy unwinding the strand, there are various enzymes such as gyrase and topoisomerase, which relieve the strain and protect the DNA strand from the stress of it being unwound above it. Now, the other strand is a little bit more complicated because on this leading strand, you only have to put down one RNA primer, and then the strand is built in the same direction as the DNA strand is being opened. This can run for hundreds of thousands of bases. But the This strand here is called the lagging strand. And the lagging strand has a complication because we have to actually open up enough that we can lay down an RNA primer, and then the replication has to take place in this direction, opposite to the way the fork is opening. And what's going to happen, it's going to run and it's going to bump up against already created strands. So you're going to have RNA, DNA, then it's going to bump up against DNA strand with its, and with an RNA primer, sorry, with its DNA strand, RNA primer with a DNA strand, RNA primer with DNA strand. So what's the cell going to do about that? First of all, it does this. It lays down the new DNA in stuttering, hyphenated portions called the Okazaki fragments. And they all each consist of a little bit of RNA as a primer, and then a short area of DNA before it bumps up against the next RNA primer. So what happens is another DNA polymerase operates on this strand, and is essentially very similar to this DNA polymerase, except that what it can do is eat out the RNA and replace it with DNA. So that's what it does, but it leaves gaps in between these strands, and those gaps are healed up by DNA ligase. Ligase is specific function is to heal these gaps by putting in the missing bonds and bases on the missing bonds on the bases. So that is the basic function of DNA replication. And remind you that it takes place extremely rapidly because all the cell functions shut down while the DNA is being replicated. Okay, so this is an energetic process. It requires cellular energy. Now, we haven't discussed metabolism to any degree, but many of you will probably know that energy in a cell is usually transported in the form of a phosphate, of a set of phosphate bonds. Phosphate bonds are extremely energetic, and they form a good temporary store of energy, which the cell uses. The most common source of those phosphate bonds is a compound called adenosine triphosphate. Obviously, tri, it has 3 phosphate groups. And the first phosphate group is the especially energetic one that and the cell, when it needs energy, it breaks off that phosphate and it uses the energy that results. Now, the process of DNA replication is highly energetic. It requires a lot of energy, but remember the nucleotides that we saw earlier. Well, the nucleotides carry 3 phosphate groups. When they're free in the cell, they carry 3 phosphate groups associated with them. So it's just the same as ATP, adenosine triphosphate. Only here, for example, we have thymine triphosphate, but it still provides a source of energy. These phosphates here are severed, and the energy is used to form this bond here. Now, one important thing to remember, this bond on the outside of the strand, the bond that forms the backbone of the strand, is extremely strong. These are covalent bonds, which are very strong chemical bonds. And remember again, look here. Here's the sugar for this thymine nucleotide. Here's the sugar here. Here's the base. And here, this is the 3' site, the 3' carbon site. And it carries an OH. So this is the 3' end. This is be the 5' end up here. For this strand here, this is the 3' end. We can see it's the 3' end because it's got that OH group, and so on. This backbone now, if you look at it, what has happened is it's running sugar there, phosphate, sugar. 2 sugars are bound together by the phosphate bond. There, there, there. Sugar, phosphate, sugar, phosphate. And these are covalent, extremely strong bonds. And same is not true of these bonds in the middle. Here is T bonded to A. Here is G bonded to C, C to G, A to T, just as we've learned. But these bonds here are hydrogen bonds. They're extremely weak. They are very, very easily separated from one another, either by heat, which we do artificially in the lab. We use this property the whole time. It doesn't happen in the cell. But in the cell, they are very easily separated by enzymes because they are so weak. And those are the processes which we have just watched happening during replication. Now, although they're very weak, DNA is a very stable molecule, first of all, because of its strong backbones, but second of all, because there are just thousands upon thousands upon hundreds of thousands of these weak hydrogen bonds, which together confer great stability to the molecule. Not only that, there's another property of DNA, and that is if these strands are separated from one another, they will realign and they will stick together with great fidelity. And we can separate DNA strands over hundreds of thousands of bases. We can separate the strands completely and then allow them to come together again. They will come back together with absolute fidelity. So this is something we'll mention again briefly when I tell you something about manipulating of DNA. Okay, so there is a difference in the replication of DNA in prokaryotes and that in eukaryotes. Remember that the prokaryote chromosome is a circle. It's a single circular chromosome, very long compared to the size of the cell, but nonetheless, it's in complete circle. And there is a little tiny point which is anchored to the cell membrane, and that is called the ORIC, O-R-I-C. And that is also in full, it's called the origin of replication. So at the origin of replication is where replication of the bacterial chromosome begins. And here's the origin of replication there, anchored to the cell membrane. And two replication forks open, one in this direction and one in that direction. And those two forks run all the way around until we end up with two rings. But there's a little problem, and that is that those rings are actually linked, like as if they were chained together. And all that happens is that an enzyme cuts one of the chains, they unwind from one another, and they are then reannealed. And what they're reannealed by? They're reannealed by DNA ligase, the ligase which joins up DNA fragments one to the other. So just remember that, and they run in opposite directions. Here's a beautiful electron micrograph of a replicating DNA chromosome, bacterial chromosome rather. And here you can see the replication forks here. They have run all the way around, and they're about to meet here to complete the replication, giving us these two different strands of DNA. So please, I just want to mention here this extra credit. I really, although it's extra credit, I really encourage you all to please do this. Please watch the movie I showed you at the start, watch that again, and then complete a worksheet which has been posted to Canvas. And that's by the end of the month, please. Okay, so we are now going to have a talk about the way in which DNA directs the creation of protein molecules. And so the first thing to remind you, go right back to the very first slides where I talked about the two kinds of polymer that we need to consider. The first polymer is that of the polypeptide. And remember that a mature polypeptide will go on to form a protein. And a polypeptide is made up, it's a polymer, obviously, and it is made up of monomers of amino acids. So get that in straight. Polymer made up of amino acids is a polypeptide, which is effectively a protein. But DNA is an entirely different polymer. DNA is a polymer. It's made up of monomers, of nucleotides. And so those are two completely different substances, nucleic acid and protein. How are we going to take the code, which we know is locked into DNA, and turn it into a sequence of amino acids, which will give us the correct protein, the correct polypeptide, one polypeptide for each sequence locked into each gene? One gene, and we're going to want to ask the question, how are we going to end up with one polypeptide? So in order to do so, we need to now look at another of the nucleic acids. And this is one that you have heard of, although you may not know the details, but this is ribonucleic acid. Go back to the beginning again, have a look at those early slides, and you'll see that ribonucleic acid is very, very similar to DNA, except that it is fundamentally single-stranded. Now, I say fundamentally single-stranded because it can, in fact, flip back on itself and base pair to itself and fold. And so that's very important for its functioning. But it is fun if we unwind it, is fundamentally single-stranded, not double-stranded like DNA. The second thing is that if you look at the carbons that make up the sugar, that is a ribose sugar in RNA. That is, it has an OH group, a hydroxyl group at the 2' carbon, where DNA has an H group. It is deoxygenated, it's deoxyribose. Ribonucleic acid is a ribose sugar with OH at the 2'. The third thing to note is that RNA contains uracil as a base. It does not contain thymine. So that is an immediate, as soon as you see a U in a sequence, you know that you're dealing with RNA, and the cell does as well. If it sees uracil in a sequence, it knows it's dealing with RNA. So there are, in fact, several kinds of RNA which we need to talk about, because there are three principal types of RNA which are involved in taking the DNA sequence, interpreting it, and producing a protein at the end of the processes. So the first is messenger RNA, or mRNA. And messenger RNA is a direct copy of the gene. So we have a gene in the DNA that is going to be copied into an mRNA copy. And that mRNA copy is going to go into the cytoplasm of the cell, where it is going to be translated. And there's a set of mechanisms which we'll examine, which translate the mRNA code, which is derived from the DNA sequence. So the second type of RNA is called transfer RNA. And transfer RNA is a really strange molecule because it consists of two things. It consists of RNA, a short piece of RNA, as you'll see, but attached to that RNA is an amino acid. Now, this is really interesting because when you think about it, we're talking about nucleic acids, right? So we've got a nucleic acid, DNA, and a nucleic acid in RNA, but somehow we have to end up with a sequence of amino acids in the polypeptide. How do we make that jump? Well, you can think about it this way. It's as if they're talking different languages, right? The DNA and the RNA, they talk nucleic acid. They don't talk amino acid. They talk nucleic acid. The amino acid doesn't talk nucleic acid. So the amino acid sequence doesn't talk nucleic acid. How are we going to get them to communicate? The same way that we get people of different languages to understand one another. We get an interpreter. We get somebody who speaks both languages. And that is the function of transfer RNA. Transfer RNA is both a nucleic acid, and attached to it has an amino acid. And you'll see exactly how it serves as a translator in a minute. The third type of RNA that we are going to deal with is called ribosomal RNA. Now, this is a very important feature of RNA in general. RNA can, as I've told you, can fold back on itself, and it can acquire three-dimensional shape. And it can also act as an enzyme. 99% of the enzymes in our bodies, in our cells rather, are protein, but a small percentage are in fact ribonucleic acids, and when they function as enzymes, they're called ribozymes. And the ribosomal RNA is both structural, it makes up the body of these little bodies called the ribosomes, and in addition, some of it has enzymatic catalytic properties which drive the process of translation. So those are our three kinds: messenger, transfer, and ribosomal. Make sure you understand the difference between them. So how do they now interact? Well, first of all, we have our sequence in the DNA, and the sequence actually is locked into units of three. Three bases together code for a specific sequence of messenger RNA. Those three DNA bases in the gene are called the triplet, and they code for three bases in the messenger RNA, which is called a codon. And each codon codes for an amino acid or an action. They can give instruction to the process, as you'll see in a minute. So we have a sequence of triplets in a gene. Each triplet codes for a codon in the messenger RNA, and those codons are translated, and they are translated by the ribosome with the intervention of transfer RNA. So this process is referred to as the central dogma. And the central dogma, this you must learn. This you must learn off by heart. DNA is transcribed to messenger RNA, this process here. Messenger RNA is then translated to protein. You can also say to polypeptide, but let's just say to protein by the ribosomes. So that is the central dogma. DNA is transcribed to messenger RNA, which is translated to protein by, and I say in brackets, by the ribosomes. This is called the central dogma because this is the basis of life for all living things on the planet. All living things use this exact same process, which evolved in the prokaryotes, but it works so well that it is still used with minor variation, relatively minor variation, by all living things. So make sure you understand and can repeat this verbatim. Okay, so what do we have to do? We have our first process that we have to understand is this process here. That is the process of transcription, reading the gene and producing a strand of messenger RNA. So that strand of messenger RNA is called the complementary strand, and it read off a DNA template strand. The transcription begins when a RNA polymerase binds to an area of the gene, which then, and the polymerase then goes on to produce a strand, a matching strand, not of DNA, but of RNA. Now that takes place in the 5' to 3' direction. The same as in replication, because why? Because it's only the 3' end of the messenger RNA strand that can have bases added to it. To have nucleotides joined, they have to join at that 3' end. And we're going to carry on that process until we reach an end. There is an instruction to end. So there's going to be an instruction to begin, to start. There's going to be a process of elongation. and then there's going to be a process of termination. And it's a relatively easy process to understand, especially if you understand what happened in replication. The difference, of course, being that only one strand is read. In any gene, only one of the DNA strands is the functional gene. It contains the useful information. What does the other strand code for? It codes for the template strand. So the useful strand is called the template. And a little bit confusingly, this strand is referred to as the coding strand. What is it coding for? It's coding for the template strand. Remembering that DNA is double and each strand codes for the other one. But in the gene, only one, the template strand, is actually contains the useful information. So I must tell you that associated with the gene, further quite far away from the actual functional part of the gene, very often, there is a little area called the promoter. And the promoter has the important function of serving as the switch which turns the gene on and off. You must realize that genes differ from one another in that some of them are required to be read off the whole time. The cell, there are certain proteins that the cell requires constantly. And those are called constitutive genes. They're continually read off. But another set of genes only switch on when they are needed. This is terribly important because if all the genes were read off constantly and were producing product the whole time, it would be chaotic and it would also be a complete waste of energy. Instead, certain genes are only switched on when they're needed and then they get switched off. There are complex ways in which this happens, but which we're not going to go into. For the moment, just remember there is this area which is a signaling area called the promoter. And that is an area where the RNA polymerase first binds. And it then moves from the promoter into the gene proper. And there is an area, there is a way for the DNA to signal to the RNA when to start transcription. So there is a sequence of DNA bases down here. When the RNA polymerase encounters it, then it knows it's a certain distance. It's usually a certain specific distance from the promoter. So the RNA polymerase then begins. It separates the strands of the DNA, and at the same time, it begins to lay down this strand here, which is the transcript. And this RNA polymerase, very complex enzyme, it does all of these things. Where before we needed all of those different enzymes, here RNA polymerase does it all on its own. This is a very narrow area, only 20 to 30 bases at a time. And this is a relatively slow process compared to DNA replication. But the RNA polymerase runs along. It reads the template strand. And look what's happening. Here's our DNA strand reading A, T, C, G, A, G, A. So what does the RNA strand read? It reads U matched to A, T, G, C, C, G, U, A, C, G, U, A, and so on and so on. Look what's happening. Where the DNA has an A, you'd think, well, it must match to T. But RNA doesn't have a T. It has U. It has uracil instead of thymine. Okay, so it has to put in a U there, not a T. What about here, though? Here the DNA says T. That's the thymine. What matches the thymine? Adenine. Well, that's fine. RNA has adenine, so there it goes. T to A to U, C to G as normal, G to C as normal. Here's another A. What do we put in for the RNA? U. So that is immediately you know this is the RNA strand, and it's reading U, A, G, C, U, C, U, 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 A. What's going to be the next one here? It's going to be A. What's going to be the next one here? It's going to be G. And notice, again, this is transcription. It's not replication. So there's only one matching strand being laid down. And this is a transcript of the DNA sequence, but now put into this other substance, which is RNA. So it's going to run, it depends on the size of the gene. Some genes are enormous, others are shorter, but it just runs right through the gene, produces a transcript, and then there is a signal for transcription to end. And at that point, the RNA polymerase falls off and the messenger RNA is liberated. And this termination signal, we won't discuss it, but there are various ways. Sometimes it's a sequence, sometimes it's a physical property of the DNA that kicks the RNA polymerase off. There are various termination signals. So what do we have? We have an initiation of transcription. Then we have an elongation period when the RNA transcript is elongated. And then we have a termination when the RNA polymerase is kicked off and the messenger RNA is released for the next set of processes to occur. Okay, so as I told you at the beginning, the messenger RNA is going to be translated into a language, a set of words, which can be translated into protein. And in the RNA transcript, 3 nucleotides together comprise a codon. And that codon corresponds to one amino acid or to an instruction, as you'll see in a minute. So remembering that we only utilize 20 amino acids. The mathematical, if you work it out mathematically, it is possible for there to be 61 codons, so actually 64 codons in general, but the 61 of them are called sense codons, and those are those that translate to an amino acid. And then there are some which are going to code for instructions. So we say then that because there are only 20 amino acids, but there's 61 codons which code for the amino acids. We say that this is a degenerate code. It doesn't mean to say it's broken up or it's nasty or whatever. It simply means that some amino acids will have more than one codon. Some have only one, but many have more than one. Some have many more than one. When we look at the codons, the possible codons, which could occur in that RNA transcript, here they all are. They form this table of 64, and you'll see that for each codon, there's going to be an associated amino acid. Now, unfortunately, in this table, they haven't given you the full name of the amino acids, but I'll tell you that when you see this in a test, I will always give you a table that gives you the full name of the amino acids. So just bear with me for the moment with this one. So when we want to look at this table of codons, remember always that this is the 5' side here. This is terribly important. It'll help you enormously to understand what is going on if you memorize that this is the 5' side. So what we have here is 5'. Let's have a look. We say 5' U, U, U. 5' uracil, uracil, uracil. That is the code for phenylalanine. There's the amino acid there. That's phenylalanine. Okay, 5' CAU. Here's 5' CAU, and that is the code for that's the codon for histidine, and so on. If the cell wanted a phenylalanine followed by a histidine in sequence, then it would have UUU followed by CAU. Now notice also, look at phenylalanine. Phenylalanine, there are 2 codons. Doesn't matter. It's fine. And sometimes the cell will use one, sometimes it'll use the other. Some, like leucine, have a look here, and you'll see leucine, there's 4. 1, 2, 3, 4, and there's another 2 here, 1, 2. There's 6 altogether for leucine. Doesn't matter. The cell can use any of them, and with the same result. So all of these here are the 61. Now, when you get this in a problem, if you are asked, well, what is the codon for serine, for example? You would have to look through here, find serine, and then be able to say, well, it's either that or that, either AGU or AGC. But what if I tell you, what if I ask you, what is the amino acid that ACA codes for? ACA. So it's 5 prime, ACA. Well, you can chicken peck your way through and find it. What did I say? I forgot what I told you now. Anyway, let me just say, I said, let's say AAU. What does 5 prime AAU stand for? You can chicken pick through, and you can say it actually stands for asparagine. So that's fine. That means you've got to look through all of these. But in actual fact, do you? Because look at how they are organized. These are all A's. These all begin with A, adenine. And here, this bar here saying first position, that tells you that. This first position, A, all those beginning with A will be in this block here. Their second letter, if their second letter is also A, it'll be in this column here. So AA is going to be in this block here. And the third position is U, and here it is there, AAU. So that's just another way of you navigating your way through this complicated table. So apart from that, there's a couple of other things to note. The first thing is this one here. And this one is AUG, and this is 5 prime AUG. 5 prime AUG is a tremendously important codon because it codes for methionine, which is very common in polypeptides. It occurs all the way through polypeptides. But it is also the codon for start. It is at this point when a particular AUG is seen that the cell will actually start to begin to build a polypeptide. So very, very, very often that first AUG is translated and the methionine is kept. Very many proteins, polypeptides and proteins actually begin with methionine for that reason. You'll see how it works in a while. So AUG is both the start codon and it is also used as the codon for methionine. It's only ever the start codon once in a sequence. Thereafter, if it occurs, it just codes for methionine. But we have some other codons which do not code for an amino acid at all. And they are these 3. UAA, UAG, and UGA. And these are the stop codons. And when in the process of translation, as you'll see, the cell encounters any of these, UAA, UAG, or UGA, the process of translation will stop. So we start at AUG, we stop at one of these 3, and in between, the cell is going to use all of the codons that it sees to bring in the correct amino acids in the correct sequence. So we've seen messenger RNA, and now we need to have a look at the other players in the process of translation. So the first one is this fantastic little molecule, which is called transfer RNA. It's a very short piece of of RNA, and the RNA actually folds back on itself several times. You can see here it's base pairing to itself, to its own sequences here. Whenever you see this like ladder structure here, you can picture in your mind that a helix develops, and it will twist the molecule in space so that the molecule begins to acquire a three-dimensional shape. And this is a rough approximation of the three-dimensional shape of the transfer RNA molecule. So this here is transfer RNA molecule, which is carrying alanine. Now, there is a transfer RNA for each one of the amino acid codons that you see in the table of codons. So there are numerous kinds of transfer RNA. And they each carry an amino acid which is specific for that codon. There's a neat little trick to the transfer RNA molecule, and it lies down at this end here. So let me just show you. Let's say this is alanine. This down here is called the anticodon. So this is CGU, and it is 3' CGU, 3' CGU. You want to tell that it's 3'. Here's the 3' end. Follow this around, and here it is here, CGU. This is important because let's go back to our table of codons and let's try and find alanine. It'd be ALA. So find alanine. Where are we? Where are we? Where are we? Where are we? We're right here. Okay. And what was our anticodon? Was CGU. We want GCA. There it is there. That's the codon for alanine that this tRNA is using. GCA. What is its anticodon? CGU. In other words, the anticodon is literally the anticodon. It base pairs to the codon. So it's base pairing to the codon for alanine. And what is it carrying? It's carrying alanine. This is how the code gets translated from nucleic acid into an amino acid sequence. It gets translated by this molecule, which carries both, which has both properties. It is nucleic acid, and it carries an amino acid. And its anticodon is specific for alanine and is specific for one particular codon, which is GCA, as we saw in the table of codons for alanine. So when we look at the messenger RNA strand, here it is here, we realize that there is the possibility now for tRNAs to come in and match each of the codons, bringing in their amino acid. And we now need to ask the question, well, how does it organize this so that the tRNA comes in the correct order and the amino acids are joined together in the correct order, in an orderly fashion? And for that, we have to look for a moment at these little bodies in the cell called the ribosomes. It is the responsibility of the ribosome to manage the messenger RNA code and to bring in the correct tRNAs at the correct time and to forge the bonds between the amino acids to form the polypeptide chain. So there's a heavy responsibility for the ribosome. It does many different things. The ribosome, as I told you, is built of ribosomal RNA, some of which is structural and some of which is enzymatic. And there are also associated proteins which build the body of the ribosome. So just let me check this for a minute. So, all right, that's just a summary. So here's what we're just going to follow along on the diagram. So here's the sequence. And you can repeat this as many times as you like to get it straight. in your heads. It's actually complicated, but there is a strict logic to it. And once that logic bears upon you, then you'll never forget it. Okay, so let's just see at the very start. This is initiation of translation. Upstream, up here somewhere, on the messenger RNA strand, there is a little message. It's called the Shine-Delgado sequence. Of course, it's coded originally in the DNA. But here in the messenger RNA, it's called the Shine-Delgado sequence. We won't discuss what it is, but its function is to inform the cell that the next AUG, the AUG immediately after the Shine-Delgado sequence, is the start codon. So here's our messenger RNA transcript, which has come from the process of translation. The ribosome is divided into two parts. There's a small part and there's a large part. And the small part is the first one to interact. As soon as messenger RNA appears in the cytoplasm of the cell, the small ribosome particles bind to it, and they bind upstream of the start near the Shine-Delgado, and they use the Shine-Delgado as their signal. And they actually move down to this first AUG after the Shine-Delgado, and the ribosomal subunit now binds to this. And once it's bound to that, it will bring in the large particle. Now, the large particle has three sites, three areas of interest. The first is called the P site, and it's this one here. So when the small ribosomal particle binds to the messenger RNA and allows the large particle to bind, the ribosome actually checks to see that this AUG is in the P site. Why? Because that is the start, and it's going to call in the very first tRNA, and it's this. Here it is here. In actual fact, I should tell you, the tRNA actually binds there, and then the ribosomal particle binds. But anyway, that's a small detail. The tRNA for methionine, in this case, has UAC. Why? Because this is the anticodon. It's 3' UAC binds to 5' AUG, binds the large particle sets, and the small ribosomal particle checks to see that everything's correct. Everybody's in the correct site. If that is correct, then the ribosomal particle allows the next tRNA to come in. So here on this diagram, the sequence of the next codon is 5' UUA. UUA. So the only anticodon which can bind to that is AAU. And that anticodon, that tRNA, brings in leucine. If you go to the table of codons and you look up 5' UUA, you will see it's the codon for leucine. The anticodon is AAU. Here's a word of warning. Always make sure, if I ask you, what does the codon UUA code for? Then you look on the table of codons. If I say an anticodon on a tRNA is 3' AAU, what does it code for? Do not look for this on the table of codons, okay? This is the anticodon. You'd look for UUA on the table of codons. So here's the second one. And now that tRNA comes and it binds there. So we've got UAC, AAU, the anticodons, AUG, UUA as the codons there. And now comes this. You must get clear in your head. Otherwise you get completely lost. This methionine here breaks off the tRNA and it joins there. it joins the leucine. It does not, this does not break off and join there. Okay, this breaks off and joins here. Now there is a set of rules for you to remember. The first is that the ribosome will not tolerate an empty A site. Just remember that. It will not tolerate an empty A site. The other thing the ribosome will not tolerate is an uncharged tRNA. That is a tRNA which has lost its amino acid. So if we move this to there, this becomes an uncharged tRNA. The ribosome doesn't tolerate that. If that happens, the ribosome moves. It moves relative to the messenger RNA by 1 codon. So now picture it this way. This is the third site here. It's called the E site. And look what has happened. This was our tRNA for methionine. It lost the methionine, which joined there. The ribosome doesn't tolerate an empty tRNA, which this one now is. And so it moves, and it moves the empty tRNA into the E site. The E site, E stands for exit or reject or whatever you like, to tell you that things there are going to be thrown out. And that is exactly what's going to happen. This tRNA eventually is going to be thrown out of the site. In the meantime, our little chain, we have a little chain, methionine and leucine. That chain, because the ribosome has moved down 1 codon to here, that is now here in the P site. And if that is the case, we're left with an empty A site, which the ribosome will not tolerate. And as a result, the ribosome brings in the next tRNA, which is going to, here is GGU. The next tRNA is going to read CCA, and that is for glycine. So remember what has happened. We joined that there. We left this uncharged. The ribosome moved to put the uncharged one in here. At the same time, it took this little chain and put it into the P site and left the A site empty. And because the A site was empty, a new tRNA came in carrying glycine. The same thing happens. Now, this chain breaks off and it joins there. Remember that. This chain breaks off. Whatever is in the P site breaks off and bonds to this here. What is in the A site? Not this way around. This is not going to go that way. This goes that way. No matter how long this chain is, it moves that way. And it will now leave this empty. We'll have 3 here. We'll have methionine, leucine, and then glycine joined. We'll have this empty. This gets kicked out, and the ribosome will move to put the empty one in here, leaving it at the empty A site again. And this is the next one that's going to be brought in. And this, I can tell you, of course, that the next codon is U, because this is phenylalanine, and its anticodon is AAA. This is just going to go on and on and on. And what we see continuously at the P site is a growing polypeptide chain. And look what the very first amino acid is. It's methionine. Methionine, and methionine, leucine, glycine, phenylalanine. What did we have? Methionine, leucine, glycine, phenylalanine. Whenever a new one is placed in the A site, the chain breaks and is bonded here. And that joining there, the enzymes that do that are ribosomal enzymes. They are ribozymes. It's RNA acting as an enzyme, and they are forming peptide bonds between the amino acids. And they're doing so in a specific order. And that order is determined now by the order of the codons. And that order of the codons was originally determined by the order of the DNA triplets. So that's how it goes on. It goes on and on and on, repeating this whole process, moving empty ones into the exit site, moving the growing chain into the P site, leaving the A site empty for a new tRNA to come in, breaking the chain and joining it to the new one, leaving that one uncharged, moving it, kicking the old one out, moving the empty one, moving the chain into the P site again, leaving the A site empty, and on and on and on. And this can go on for hundreds of thousands of codons. Some of the peptides, polypeptides that are going to be formed are absolutely enormous, absolutely enormous. And as they leave the ribosome, they begin to be processed in various ways. And the polypeptide will be processed to eventually end up forming a mature protein of the correct shape, the correct folding, the correct properties, and everything else. Now, eventually, the codon sequence will include a stop codon. It will be any one of the 3 stop codons that you see in the table of codons. In this case, it's UGA, could be one of the other ones. And but when that stop codon is placed in the empty A site, no anticodon matches that. No tRNA matches this stop codon. Instead, a set of proteins is brought in called the releasing factors. And these cause the whole complex to fall apart. The protein, the polypeptide rather, is released, the ribosome halves are released, and the last amino acids are released. By the way, the amino acids that are uncharged, they can be reused, and they're reused quite extensively. They just go back into a cellular machinery that recharges them, puts their correct amino acid back on them so that they can be used in another translation event. Ribosomes are unaffected by the whole process. They, in cellular terms, relatively long-lived, and they do this thousands and thousands of times before they eventually are recycled or whatever. So there is a movie for you on translation. Now, I must just tell you that the movie is actually about translation in eukaryotes. And in eukaryotes, translation is a little bit different because in eukaryotes, of course, all the genetic information is contained in the nucleus, or most of it is in the nucleus. So the messenger RNA strands are produced in the nucleus, and they actually have to be exported from the nucleus into the cytoplasm. Before they are exported, however, there are various manipulations that are made on eukaryote messenger RNA before it becomes functional and can be translated in the cytoplasm. We're not going to go into the details of that. We are more interested in the prokaryotes. But in any case, the principles of translation that you'll hear about in this movie are basically the same. So please make sure that you watch that. So there's what I've just told you about the difference between eukaryote and prokaryote has another implication. In the prokaryotes, there's no nucleus, of course. And the messenger RNA is produced directly inside the cytoplasm. So the DNA strand is in the cytoplasm. As transcription takes place, a messenger RNA strand is produced. And as soon as even a tiny bit of messenger RNA appears, ribosomes will glom onto it, and they will start the process of translation immediately. And this produces a phenomenon where this is a marvelous piece of microscopy here. And this is actually a DNA strand here, there. And what is happening is that is being transcribed. You can't see the transcription machinery, but you can tell that there is a messenger RNA strand because this here is a whole chain of ribosomes which have stuck onto the messenger RNA and are busy translating it as it's being produced. So the first thing to ask is in this, well, which is the oldest messenger RNA strand? That's relatively easy to answer. It's this one here, the longest one. Okay. But what the other thing to ask is, you can't see protein here. All you can see are the ribosomes stuck to the messenger RNA. But the proteins are trailing off behind these ribosomes. Where do you think, where's the longest polypeptide? Is it this? We know this has been on longer, right, than that there. This is just, this, the RNA polymerase is just busy, this RNA polymerase here is just busy producing its first bit of messenger RNA. So it's not going to be there. It's this strand here, but where on this strand would we look for the longest piece of polypeptide? Over here or over here? And the answer is here. Because this ribosome here is just following along as messenger RNA is being produced. This ribosome here has been stuck on here longer than anybody else. The messenger RNA trails behind this one, the original, and other ribosomes stick onto it as it's freed from this particular ribosome. So if you have a look, here's the interpretation for you. Each of these little strand of beads represents a messenger RNA strand. Each of messenger RNA strand is being produced by another RNA polymerase. And this also gives the lesson that from a single strand of DNA, we may have many transcription events running at the same time. And on any messenger RNA strand, we may have many ribosomes performing translation. So it's a very efficient process. As soon as that gene is switched on, its product is produced in abundance very rapidly. Okay, so I think that I am just going to leave that there for today. And then I'll see you again on Monday, of course. Please make sure you watch the movies here. And please pay attention also to the piece of homework that I included there for you. Okay, I'll see you on Monday.